vaccine. It's sometimes it's with dose one, and then it doesn't happen with dose two, and it's been the same. It was the same vaccine, and for some, it's created apprehension about getting booster doses and so on. And I think that's something where it's an intervention we cannot control because it's rolled out governmentally, and the patients obviously have to get protected, but the biobank can maybe help us understand what's happened before and after in relation to these symptoms, because I think that is really needling something. But I would be open to hearing of more.
The idea of persistence. So the idea of SARS-CoV-2 being persistent is one, but then you have
SARS-CoV-2 are anemia, right? And so is there a role, a distinct timing for antivirals that could then perhaps protect against long COVID? I think that's a really important question. Dr. Rigi. Oh, we are on mute.
Oh, thank you. I just wanted to point out that our treatment strategy has completely changed from wave 1 to now. So the patients I'm following up to 18 months, they are from the first wave, where we almost got told not to use steroids because it was a bad idea. And we were using hydroxychloroquine and lopinavir, ritonavir that definitely were this, for example, as I'm researching how disrupted they were on the gut microbiome, they definitely so probably also the consequences of the type of treatment are really important. Now, basically, we're only using steroids and trying to get something else in the acute patients, some antivirus, resuming remdesivir, but not really that much, and also anti-inflammatory drugs that have a very long half-life that have been used, for example, in autoimmune disease. But these drugs can have weeks of half-life, so they are in the body for a very long time, acting as probably saying anti-inflammatory is even trivial because they have complicated mechanisms of action. And we abandoned the idea of doing the profile of cytokines to understand if the cytokine storm was occurring or not. But all these elements about treatment and treatments that are not like antibiotics that have short half-lives, but they can impact on these patients for a long time. Is a confounder, but probably needs to be followed up. And I'm also sorry, but I have to leave now. In advance, I apologize, but unfortunately, I have to go. And I just want to thank you, everyone, for all the inputs and the discussion.
Thank you so much, Dr. Ryu. Your participation was really very valuable, and I really look forward to discussing with you offline.
Thank you. Goodbye. Bye.
All right. So, in fact, with that, I'd like to transition now to slightly more molecular aspect of the discussion, where we're going to talk about prevention and treatment of COVID, but also emerging viruses. And some very interesting approaches have been raised, including using population-based approaches for drug discovery. The first question I want to throw out, it's a little bit, general, but how do you see the need for, I'm going to use a buzzword here, but precision medicine, whole genome sequencing on individuals, population, genome variants, and the interplay with these types of drug discovery approaches? I'd like to direct the question to Dr. Tyers and Dr. Granvaux. You could choose who goes first.
I vote that Natalie goes first.
Well, that's a very good question. Well, I would say that
do you think it's essential? I guess let's start with that. It's essential to have this information for us to, especially if we're going to go with, you proposed, the idea of a drug that sort of, leverages host mechanisms so that we could deploy it right in any viral. So we're not depending on the highly fickle virus. Okay. So, I mean, would that change maybe if we had this information ahead of time, would that change the choice of antiviral we would use in certain subpopulations, even if we just start with different ethnic groups?
Well, I think that's it would be important if we know who are the vulnerable people. As you say, there is a population which we know will be, if we go back to SARS-CoV-2 at the beginning, having such kind of molecule, we would have targeted them to elderly people, definitely. It doesn't need to be in all the population, but it would have been very, very useful. Do we need more molecular information? when we use those one? I hope not, because that would be counterproductive, But we will still need, on the top of having these antivirals, we would need to have the information, the molecular information, to slowly move on to more specific targets, more specific antivirals. I don't think that having the broad range on the long term would solve all the problem. It would help us to wait for something more specific, especially vaccine or specific antivirals, which will be probably more potent. But I think, to me, I see that as a sequence, and it's complementary approaches. Where the molecular testing, I think the molecular testing is key when you have virus targeting antivirals, because especially now, so Pfizer has shown that Paxlovid works with Omicron, but it may not work. We never know. If there is a mutation in one of the enzymes that is targeted, if the catalytic site is changing, it's quite rare, I have to say. But we have had so much surprise with this virus that I'm not excluding any possibility now. So we need to know what is there, to know whether, and have possibility of rapid testing if the antivirals, the tools that we have, are still working with emerging variants or new molecules. So I think that's where the interplay between the two, I would see them. I don't know, Mike, what you think about that?
Well, the reason I hoped you would go first, Natalie, is that I think it's much more in your ball court in terms of the innate immune mechanisms, for example. And my understanding is that there is some signal in GWAS studies, but I think most GWAS studies, it's only a minority of the say the severe cases can be explained by polymorphisms and, say, innate immune genes. So I think it's going to be very complicated. And I think this also gets to the issue of what is, in the long term, what kind of therapeutics or antivirals will one need. And to go back to your point about HIV as a comparison, I think combination therapies are going to be very, very important, not only for improved efficacy, but to mitigate resistance. And I think the size of, the extent of infection across the human population has created an enormous reservoir, and the propensity for resistance is really quite worrisome. And so getting ahead of that early and taking lessons from HIV, I think, is very important.
I completely agree with the need for combinations. I think that's very, very good point. And I wonder, is it combination of antivirals, or is also molecules that can prime the immune systems in certain directions, things that we could leverage? Two of the mechanisms, yourself, Natalie, mentioned is, autophagy and reactive oxygen species, which are intimately related. I mean, do you think, I think back at, my pre-COVID interest in patients with chronic granulomatous disease, right? Who they do not make their phagocytes don't produce reactive oxygen species, but presumably the epithelium does. Yet paradoxically, they have a hyperinflammatory state because they have defective autophagy. How do you see that interplaying, the relationship between reactive oxygen species and autophagy in the context of viral infections?
Well, that's exactly what we work on. It's the problem with, and I'm actually my background is from CGD also. That's where I've been trained during my PhD. So that's where my redox part is coming from. And
this wasn't on purpose, by the way.
I didn't know you worked on that. So the thing is that's exactly what you describe is exactly the reason why we move to identifying the cellular target of reactive oxygen species. Because if you stay at the level of just looking at global oxidative stress. First, you miss some part of it, but also where the ROS are produced from which cells, at which time, completely change their impact. So, so when we switch to epithelial cells, actually the enzyme that is present in phagocyte is also present in the epithelial cells, but they have other oxidase, like the DOX2 I mentioned. So basically, they are not relying only on one. And the targets are different. So I think that I see that a bit like, and I'm actually writing a review on that right now. I see that like we learn about phosphorylation. You know, at the very beginning, when we learn about phosphorylation of protein, people were using phosphotyrosine, phosphoserine, phosphothreonine antibodies as marker of oxidation happening. But now we know which site is oxidized, is phosphorylated by kinase, and many therapeutics we use target the kinase, but for a specific site, you know. And I think for me, for the oxidative stress, we need the same knowledge. We need to know where the target, and we need to be able to use that first as biomarker. That would be awesome to be able to do that. There are some challenges, but also to be able to modulate the oxidation of specific sites. And coming back to autophagy, the interplay between the two is really cell type specific, which is what makes it complicated. You know, if you take the neutrophils or the macrophages, you know, if you don't have the NOX2 pathway, you don't have this happening. But in the other cell type, it's not completely dependent. So it's really, the complexity is the cell type specificity, to my opinion. So that's why, speaking about respiratory viruses, we study the interplay between the epithelial cells and the neutrophils right now, because they communicate together. But what we have to be very careful of is if we target the oxidation processes or the autophagy process in the epithelial, with the aim to change the epithelial cells reaction, we may also impact on the neutrophils and the macrophages that are in the lung. So we need to make sure that by helping one cell type, we don't trigger an inflammatory process in the other one, you know?
I completely agree. And I guess I would ask the question in the other direction as well. You know, what is the impact of the emerging neutrophils that emerge in the chronic and the acute inflammatory response and their imprinting on the epithelial cell, which then could also alter the mucosal environment, which houses the microbiota.
Yeah.
And, you know, so the community of microbes that inhabit a given location on the body. And that microbiota also makes reactive oxygen species.
Absolutely.
So I think that it's not simple, but these are, I agree with you that they're very high, big ticket items that have to be looked at in the context of even viral infection. Now I have a question for you, Dr. Tyers, about your CRISPR screens, which I think is absolutely fascinating for those of us who don't do this every day. When you find interesting targets, I mean, my concern would be, well, if you start to like affect some of these targets, what other functions are you going to affect? And how do you validate these targets? You know, how do you get them out of the in vitro setting and into something that resembles a human?
Yes, well, I think that's a key, if not the key question. You know, and the problem is that certainly discovery of small molecule therapeutics is a 10 to 15 year timeline, even, you know, when you start with a very well-validated target, and we don't yet have well-validated targets. So, you know, so the easy part is actually doing the screens in culture. Then, you know, the next step is the preclinical models with, you know, say different, you know, mutant mouse backgrounds or other transgenic animal systems, even just to, you know, get to the point where you can start to validate small molecule inhibitors. So I think it's a very long road, and it's for the long game. I do want to pick up on one point that Natalie raised, which is cell type specificity, and this is a real, you know, a confounding factor in interpreting the genome-wide screens. I think at this point I can pretty safely say there is no magic bullet. i.e. no magic target, because depending on the cell type, and even just the tissue culture, nuances and details, screens, even done in the give different, will give a different readout. And Jason Moffat in Toronto, who's done a lot of these screens as well, has a preprint basically saying the only common target is ACE2 across all screens in different cell lines. So that's very worrisome. The other very important point that Natalie alluded to is the genetic landscape differs between different cell types. And I think that's the explanation for this, is that while at a process level, it may be important for viral resistance or sensitivity, at a genetic level, that can be very different between different cell types. Either rate-limiting genetic factors, which is what these genetic screens give you, are different between different cell types, even though the process, may be universally important for the virus. So I think this is the hard thing about kind of host-directed antivirals, the genetic complexity, and that has to be weighed against the advantage that if you can identify a host target and develop a small molecule therapeutic against it, you're way ahead of the curve in terms of resistance. So I think the viral targets are easier, they're more prone to resistance. The host targets are more difficult and maybe more resistant, more recalcitrant to the emergence of resistance. So yeah, I think it's this is a very sort of going to be a long process, but I think now that this sort of genome-wide CRISPR screens are being used very broadly in virology, there's a lot of information coming out. Different virus screens have been done, and I'm hopeful that there will be themes that will emerge that will play into the pan-viral concept that Natalie elaborated in her talk.
Yes, and I think, as much investment, I think this is the type of forward thinking that we need to have, and we need to have in place to be ready in the future. So we have Dr. Plobani, who's has to go, but we thank you very much for your participation and your input. Very valuable information on diagnostics and monitoring. Thank you.
I have to thank you, and I really hope to have other opportunities to meet you in person, possibly, and to have a much more cooperation. I love Canada, and I love much more Quebec. So I will be in June. I will be in Canada, or better in Quebec, for a meeting. Hopefully.
We will be happy and most openly willing to welcome you with open arms in our land, and likewise, we hope to visit you as well.
Thank you, Professor Plobani.
Thank you.
So yes, so I think it's well worth the investment, because we have to innovate definitely in this direction. Now, I was intrigued by the engineered probiotic yeast, and I guess I have a very basic question. How long does colonization with such yeast last?
Yeah, so that was one of the key things we've looked at. So we've been looking at both kind of oral. So one of the advantages, I skipped over it very quickly. One of the advantages of this probiotic yeast is it survives passage through the stomach and the GI tract, and will effectively populate basically the colon at all tissue levels. So you could argue it both ways. What do you want? Do you want a probiotic that's going to permanently colonize and produce a therapeutic in situ over the long term? Or would you rather just have transient colonization, so it's a bit more like a drug that you can maybe take it every 3 or 4 days and that would suffice? It looks like the latter is the case. So both in the colon and in the nasal epithelium, I showed the decay curve for the nasal epithelium. It's after basically 3 days, yeast are gone, so they don't permanently colonize the nasal mucosa. In the colon, the persistence time is 1 to 2 weeks. So the other thing I didn't mention, at least for the gut microbiome, is that Saccharomyces cerevisiae, sort of that clade of yeast, is part of the natural microbiome. So it's not like we're adding something completely different. We're just adding a yeast that you can find in the microbiome that happens to be making, recombinant protein. We're also exploring small molecules produced by the yeast in the same way. So yeah, it's an absolutely key question, and it looks like the half-time is a small number of days to a week or two, depending on the tissue.
I think that's very interesting. I would agree. I think one would favor an intervention that's transient, that you can control.
Yes.
And and it potentially could get influenced if the patient then has to go on immune modulator, steroids, antibiotics, antifungals, etc.
Yep. Absolutely. Yeah.
Do you have any evidence, because you did mention the microbiota, of what the impact of this colonization is on the composition of the rest of the microbiota? And also, do we have evidence of gene exchange? How does that, we know that microbes talk to each other.
Two other great. I have to put you down for a review or for a grant if we submit it. I think so on the genetic exchange front, that's one advantage of yeast, is that there's no horizontal gene transfer. So I think, unlike bacterial probiotics, where horizontal transfer between bacterial species is very prevalent, that does not occur with yeast. So that's one advantage. And then in terms of the impact on the microbiome, those experiments are in progress. We don't know yet, but we're looking at it. Not so much in the context of the nasal microbiome, but the gut microbiome.
Excellent. Exciting. I think that's very encouraging. Actually, I'm even more intrigued than I was before. Do you think some of these living therapeutics could also have an impact on response to vaccines?
Good question. I had not. We had been thinking we've tried experiments using them as vaccines, and the data is mixed. Depending on the context, there can be a weak immune response. There's one or two published papers suggesting that oral administration, actually of fixed yeast, can elicit a very strong immune response. So I think it's very early days for that. So yeah, I think. We'll see how it all plays out.
Interesting. Okay. So at this point, do the panelists have any burning questions they would like to ask each other?
I think I'm burned out.
Well, what I would say is, I from what I hear, is an amazing infrastructure to monitor not only COVID but emerging diseases, an infrastructure that has pivoted in response to the pandemic, and that is evolving, and that seems very, very dynamic to me. And I guess I have to ask, a $1 million question in this setup. How can we merge the data you're collecting and your infrastructure with some of the expertise that we have here to sort of create something new? Do you think it's possible for us to sort of work together, to have access to data, to samples, to be able to exchange?
Of course, I think it is possible. I mean, it's a challenge to merge and to work together, but I think it's what we need to do to learn from what we have faced, what we have collected, and see how to read a huge amount of collected from different actors, hospitals, long-term care, GPs. So we have really a huge amount of data that we need to read them in a complete way. And I think we need to work with other countries, other ways that have been collected. We're still doing it. We're already doing it with our colleagues, with our professionals that works in Veneto region and in other countries.
Yeah, yeah, and so I think this is definitely an important first step in opening the dialogue and the pathway of communications between different groups, because I really do see a complementarity with all the wonderful work that you've been doing in the Veneto region. It's really fascinating. So again, last